Hello, I'm Diana Northup from the University of New Mexico, and I and Kathy Loy, who's here with me from SUNY Plattsburgh, will be presenting on who will be dead after we save the bats. This is a project that we've been doing for the last couple of years in conjunction with several people from the National Park Service, who you'll see here: Ivan, Alice, Eric, John, Katrina, Rick, and Jason are all NPS folks. And then the first people listed, Anna, Christopher, Evan, and Shane, are students in my lab. And they're all part of this big project to investigate what we're going to tell you about today. Kathy's going to lead off.
All right, well, you're all familiar with white nose syndrome, which has killed millions of hibernating North American cave bats since the winter of 2006, 2007. And this is close to home for me because this was first discovered in New York State. The fungus, Pseudogymnoascus destructans, is an invasive introduced from Europe. And you can see in this picture of the bat, the classic white nose from the growth of the fungus, and then the growth also on the membranes of the wings. And there's a lot of work being done to try to find ways to control the fungus and reduce the fatality among bats. All right, a little background on ultraviolet radiation, because we're using ultraviolet C radiation to try to control the growth of the fungus in the environment. And ultraviolet is part of the electromagnetic spectrum that comes from the sun. And down here we have the example of the portion of the electromagnetic spectrum, visible light in the middle with the Roy G. Biv going from right to left, red to violet. Below red we have infrared, and above violet we have ultraviolet. Now notice the wavelengths down here, shorter wavelengths on the left and longer wavelengths on the right, and there's an inverse relationship with energy. So the shorter the wavelength, the more powerful the radiation is. And we're all familiar with ultraviolet radiation in that it causes sunburn and skin cancer, particularly melanoma, also contributes to formation of cataracts and the aging of skin. Now, what you're not used to is that there's actually 3 different varieties of ultraviolet radiation. UVA, the ones that cause the effect you're with, but UVC, which has length and is the most powerful form of ultraviolet radiation, luckily for us, doesn't reach the surface of the Earth because it's completely blocked by the ozone layer. So organisms have no adaptation to UVC radiation because they never see it. One important point about all types of ultraviolet radiation is that they're fairly easily blocked. That's why when you're getting a tan, you get tan lines because it doesn't penetrate through your clothing. You can't get a sunburn in your car because it can't get through the windshield. And again, of all 3, UVC is the most easily blocked. Okay, we have a couple of objectives of our work. The first one, as Diana mentioned, is will UVC treatment have the potential to kill native cave bacterial species? And then what we're going to be talking on today is a part of that research where we are identifying the effects of UVC treatment on the growth and survival of cultures of bacteria isolated from caves. As Diana said, this is in partnership with the National Park Service. We're doing this work at 3 different caves across the country. Of course, Oregon caves up in Oregon, Lava Beds National Monument, which is in Northern California up into Oregon, and Mammoth Cave in Kentucky. And I should point out that these are 3 distinct types of caves. Oregon cave is a marble cave. Lava beds or lava caves, and Mammoth Cave is a typical karst cave. So we've got a good variety of types of caves here. Here's an example of our sampling in Oregon caves. So you can see the cave, the main entrance is up at the top, and tourists go through in a one-way direction on the blue trails and exit the cave. And then down here at the bottom of the cave in orange is an off-trail portion of the cave where, which is going to have less impact from people. Now we have 3 sites identified in the cave, and these are picked because these are areas where bats normally hibernate. So if they are carrying the white nose fungus, you'd expect to find it there. So that's it for this slide. Now here's an example of sample collection at Orca. First of all, notice that the people doing the sampling are wearing a lot of personal protective equipment, including face masks, Tyvek suits, and gloves, which they're going to be changingly. And they're doing this not to protect themselves so much, but to protect the microbes in the environment. Of course, these are caves that have bats, so there's the potential for histo, so it's a mutual protection. Now, what we are showing here is an area on the walls, and we've also sampled from the floors and ceiling. But you see these 3 rings that are made out of PVC tubing, and they're divided by wires into 6 quadrants, or 6 sections, and we call these pie slices. And when we sample, we take a sterile cotton swab, moisten it in sterile saline, and then rub it across the entire surface of one of these slices of pie. And that is 1 sample. Okay, so we're going to use each of those swabs to plate on different culture media. We used 3 different types of culture media, all of which are fairly low energy to more closely mimic conditions in the cave. And in addition, 2 of the 3 had rock dust in them, which is rock from that particular cave, to create a more homey environment for the microbes and provide them with some nutrients that they might be needing. So we took these 45 swabs, 3 from each of the 3 sites in the cave, and swabbed them across the entire surface of the plate. So this was done in the cave, and then the plates are brought out, put on ice, and shipped to Diana Northup's lab in New Mexico, and incubated at 8 degrees centigrade to mimic cave temperature. And the initial incubation took anywhere from 2 to 8 weeks to get a good growth of the microbes. And you can, again, if you look at the plate, you can see all these growth of individual microbial colonies. They might not look all that different to you, but when you look closely, there's going to be differences in size and shape and texture and color. So we can discern a good variety on each of these plates. So we selected for a good variety of morphologies and subcultured them on the same medium that they had been isolated from. And it generally took 1 to 2 weeks for each incubation. And it may have taken many cultures before we finally got to the point where we thought we had pure cultures. So once we had these organisms isolated, the DNA was extracted from each colony, and then the 16S ribosomal RNA gene was sequenced. The 2 reasons for that: 1 was to determine if the pure cultures were actually pure, and secondly, to identify what bacteria made up each individual colony. Now here's some example of what we found, an overview of what's in the cave. So of 53 cultures that were sequenced, 47 were pure cultures, and that's what you're seeing here. And this is a pie chart that breaks down the different species the different genera of the bacteria. And you can see that the biggest part of the pie was taken up by Arthrobacter species, which is a type of actinobacteria, and then Flavobacterium, which you're probably not familiar with. That's an example of a Bacteroidetes bacteria. Diana?
And I want to point out two additional genera in here that are of interest that are related to white nose syndrome and its treatment. So besides UVC as a treatment, we've also been investigating over the last few years the use of bacteria that actually live on the bats. And so the Streptomyces is a genus from which we get a lot of antibiotics for humans, and we found that it's also quite prevalent on bats, and we're able to culture it from the bats, and in the lab have tested it against the fungus P. destructans that causes white nose syndrome, and it will kill the entire plate of that. Rhodococcus was actually investigated by Chris Cornelison, and he did both lab and out in the field in a private cave, looked at the ability of Rhodococcus to kill white nose syndrome in these different situations, and it was very effective at doing that. So these bacteria represent both the native cave bacterial populations, and also some of them are of interest for other reasons. So we take these cultures, and we're doing 100 from each of the parks. We're only going to be showing you results today from Oregon caves. The others are still in process, as are part of the Oregon Caves 100. So we're going to show you our first results from this. So once we have these cultures, we basically do a fresh subculture, and then when it has grown for 6 days, we basically take a new culture loop of material off of it and inoculate clean new plates to subject to UV radiation, UVC radiation. This is done in a machine you see in front of you on this slide, and it's a UVP crosslinker. And we do 3 replicates at a time. We do a control, which is put into the machine, has the isolates on it, but no UVC is added. Then secondly, we do 3 replicates that of the same isolates that are put in here and subjected to 5 seconds of UVC, which is equivalent to 25 millijoules. And then thirdly, we do another 3 replicates of those isolates and subject them to 20 minutes, which is equivalent to 100 millijoules. That gives us plates that have been exposed to the appropriate UVC radiation. We immediately wrap them in aluminum foil and put them in the 8-degree incubator for exactly 6 days. At the end of that time, they are taken out and photographed. And my husband, Kenneth Ingham, has set up a sort of little photo studio in the lab that has set up to basically illuminate the cultures uniformly so that we don't get various bright spots on it, and it's much easier to analyze if we do that. And those images are then processed in Photoshop and then quantitated in a program called ImageJ, which actually measures the area that has growth on the culture plate. So here's what it looks like. So what you see here are one set of replicates. Notice that there are 4 different isolates on here. So here's the first 4 of the 12 that I'm going to show you results for. Notice that the color and texture looks different from quadrant to quadrant. That's because these 4 are different. This one had no UVC. The middle plate has the same 4 isolates inoculated at the same time as the control. And you'll see that the upper 2 have a reasonable amount of growth. The bottom 2 either have none, or very little growth. Then we go over to the 100 millijoule, and you'll see the top two that had quite a bit of growth with 25 millijoules have quite a bit less with 100 millijoules. Not that much difference in what we see in these two quadrants. So this is a way that we can actually quantify how much killing off of the isolates the UVC does. So if we take the first 12 different isolates that we have subjected to UVC and run them through ImageJ, we are able to produce a graph that shows the impact of the UVC. Notice that the y-axis only goes up to 50%. That's so you can better see the results. And the 25 millijoule plates basically had only 20% survivorships. It looks like the 100 millijoule had about 18%. So you see a great reduction in the growth of these cave bacteria when they are hit with UVC. This allows us to evaluate if they use UVC as a treatment for white nose fungus and to kill it in the caves, that it is going to have an impact on the microbes in the caves. And it's pretty effective at killing several of the cave microbes in particular. Now, this could lead to a very negative outcome of altering the native cave microbial communities, but there's some caveats to this. We know that we're only able to grow at most 10% of what actually is out in an environment. So our study is not representative of everybody who's there. So further study is needed, and we have a phase 2 where we're going to look at also at recovery and whether that happens too. We would love to have questions from you. You can email us, dnorthup@unm.edu or lavoykh@plattsburgh.edu with your questions. And we'd like to end by also thanking our sponsors and also all the people who contributed a lot to this. So in particular, the staff at Oregon Caves National Monument and Lava Beds National Monument actually did the initial collection in the caves. And one man in particular, Ivan Yates, actually went to all 3 parks and supervised the collection so that we have a uniformity of how the samples were collected. I'm really grateful to all the people in my lab, all the students and staff who were part of this project. We have an entire cold room full of cultures. We have something like 3,670 cultures, from which we are whittling it down to the 300 that will ultimately be tested. Really grateful to the Park Service who provided funding through the Colorado Plateau Cooperative Ecosystem Study Unit, and to my husband for all the help in making the photography work so that we could actually measure the difference. So many thanks to all these people, and we thank you for your attention to this presentation and hope you're getting an insight into who will be dead once we save the bats.
Thank you.